Hi, everyone. Thank you for being here. It's a great pleasure for me to present today. So a brief overview, basically, of my research aims and how they relate to my current postdoc project that I'm currently undertaking, as Antonio said, at the University of Bern in Switzerland, working in the Museum of Natural History and collaborating with researchers of the University of Cambridge. So as I will detail in this talk, my research interests mainly concern the evolutionary and adaptive factors related to bone inner structure in tetrapods, so basically vertebrate with leaves. As Antonio said, I come from paleoanthropology, so basically my main focus is still on primates. But for my PhD, I studied mammoths, so not only primates, but also sloths, perezosos, and other species, and now I'm working on birds. But as I will show you here, as I will show in this talk, basically the methods and the theoretical framework is basically the same across these taxa. So the first logical question may be, so what do you mean with bone inner structure? So think about taking a bone, a skeletal element, and dissecting it virtually, for instance, through micro-CT scans. And you can see internally the repartition of bone tissue. So basically, you see this internal repartition of bone tissue at this anatomical level. This is what I mean when I talk about bone inner structure. So we know that a multitude of factors basically affect the diversity of bone inner structure across vertebrates. So for instance, we have allometric aspects relating to scaling patterns only related to size. But we also have phylogenetic factors. So as Darwin told us, species tend to resemble themselves, other species, depending on the evolutionary history, so how recently they diverged each other. And this is, of course, a factor affecting the diversity of phenotype at any level, including this one. We also have, of course, ontogenic factors, so related to developmental patterns of single species-specific developmental patterns. And ecological factors, so relating to how the species interact with the environment, for instance, through locomotor behaviors. And this latter factor is the the one I mainly focus on, as I will show you better later. The thing is, once we find a relationship between morphology and ecology, so let's say locomotion, for instance, so once we find a model of correspondence between a morphological aspect and locomotor aspects, we can use this covariation, this correlation, to infer paleobiology, so locomotion, for instance, in extinct species, for which we only have morphology. So this is the key tenet of functional morphology. And basically, this is the main factor where on which I focus. So when we talk about how internal bones, inner bone structure relates to ecological aspects as locomotion or biomechanical aspects, as I told you, we should remind a key tenet in functional morphology related to the thing I just told you. So basically, we know that the bone structure tends to respond to mechanical loads, to mechanical loadings to which the skeletal element are subject during life. This concept has long been termed Wolff's law, named after the German engineer who first theorized this relationship. Today is better referred to as bone functional adaptation, but the thing is, basically, the important concept is that every time a bone is subject to stress, as a strain, depending on locomotion, so biomechanical aspects, it triggers a sensory perception, which leads to the activation of osteoclasts. If the strain increases, osteoclasts are responsible for the deposition of new bone tissue. So basically, it brings to bone growth. On the other hand, once loadings are decreased, so every time we have like a decreased loading magnitude or biomechanical load, we have the activation of osteoblasts, which are instead responsible for bone resorption, so basically having less bone tissue. So you can understand this dynamic equilibrium brings to the bone to partially register the mechanical environment. And we should be somehow be able to read bone morphology, because the bone morphology in a way records biomechanical history. So just would like to be more precise when I talk about bone inner structure as an anatomical level. Although with several adjustments in the decades, today is still well established the Peterson classification of bone levels of integration. So I will not go too much into details due to time limits, but you can see here that we have, depending on different magnification scales, okay, 4 orders of structures. They go from general morphology, the one we can see to the naked eye, and this is the one most commonly studied, for instance, through 3D geometric morphometrics or indices, metrics. You can just describe the bone without any quantitative metric, but you can go down to the molecular level of integration of bone phenotype. I will show you that I mainly focus on the first 3 orders. I do not really study a cytological level of integration, but as I will show you in my PhD, I worked on structural trait, that can be appreciated only with few microns of resolution. And another important aspect is my main focus on some skeletal elements. So since, as I told you, I mainly aim to understand relationships between bone morphology and ecological aspects as locomotion, the postcranial bones, so everything but the head, basically, but the skull. The postcranial bones are particularly suitable since they directly interact with the environment, for instance, through locomotion. But they are also involved in weight bearing, so in allometric patterns. But also they are also affected by evolutionary patterns, so by phylogenetic signals. So we can have like a summary of all the factors affecting bone morphology, studying the postcranial bones. And specifically, so far I mainly focused on long bones, so the key elements of the appendicular skeleton, so without the axial skeleton, so basically limbs. So if we look at the internal structure of a long bone, here exemplified by the humerus of a tree sloth, we can describe different regions in both the extremities that are called the epiphyses, as you probably know, and the long central shaft named diaphysis. So you can distinguish the external compact layer of bone called the cortical bone or cortex from the internal less dense region lining the medulla. So in this region here, in this tube, let's say along the central shaft. And within the epiphysis, the life is filled with marrow and bone, but of course marrow after death decomposes, yeah, and so marrow decomposes, and so we have like bone and void, empty spaces, and these builds in the epiphysis the so-called trabecular bone. That I will show you and I will talk about better later. So another question maybe now, how can we access bone internal structure? So the traditional approach, but still valuable and used today, is the production of thin sections, allowing to describe specific features through microscopy, as you can see in these beautiful images. One drawback of thin sections is, of course, that it is a destructive technique. So if we have like a very rare specimen that we cannot destroy it because it's like a fossil, for instance, it is not the best to use thin sections. That's why we alternatively use virtual imaging, like widely spread microcomputer tomography, but also synchrotron, for instance, because of course the destruction of the specimen is not involved in these imaging techniques. Introduced several decades ago, they allow us to study the 2D, but importantly also the 3D distribution of bone within the studied skeletal elements. And this is the approach that I mainly adopted so far. So briefly, which are the characteristics that we describe and ultimately quantify in diaphyseal along the central shaft, as I told you, and epiphyseal, as I will show you later, the extremities of long bones. So the thing is that the diaphysis has been assumed to be mechanically comparable to a tube, or better to a beam. So it leads to use the so-called beam theory. So this mathematical engineering treatment is widely used in engineering, as I said, and theorizes how beams should be built to optimize different mechanical loadings to which they are subject. So since they are long elements, meaning that they have one size exceeding the other two. So you have basically a long element. Yeah, they are subject basically to 4 types of loadings. 2 of them are axial, so occurring along the long axis, compression and tension, depending on the direction of the loads, and we also have torsion and bending loadings. So the important thing is that it has been shown that if we study a cross section, so a bidimensional, 2-dimensional cross section along the beam, so the diaphysis, we can have basically all the information we need to know on how the bone is optimized to respond to loadings. And so quantifying some parameters in the so-called with the so-called cross-sectional geometry, we can basically understand the loading regime of the long element. So these are the kind of parameters you can quantify in diaphysis of long bones. Talking about another inner bone anatomical level, that is the epiphyseal inner structure. So as I showed you already before, that especially long bones within their extremities have this peculiar spongious structure. It is also called spongious bone or cancellous bone. So we have this network of bone struts named trabeculae, and as I already told you, their features were related to ecological aspects, as locomotion, and of course related biomechanical aspects. So the Wolff's law, the law I mentioned before, the principle I mentioned before, has been tested several times on trabecular bone, for instance, training animals to perform specific locomotor habits, and then testing if the resulting trabecular bone somehow responded to the loadings deriving from these behaviors. And although with mixed results, with some controversial aspects, but we can say that in general there is a correspondence between trabecular bone structure characteristics and locomotor aspects. Here again, I will not go into detail into the single parameters you can quantify, but in general, you, for instance, can quantify the degree of orientation of the struts that can inform on the main direction of loadings acting on the epiphysis of long bones, or you can quantify, for instance, the density of trabecular bone informing on the magnitude or the frequency of the loadings. So there is like huge literature on this correspondence between some characteristics and locomotor aspects. It is important to remind that, as I already mentioned before, all these traits are strongly affected by allometry. So it means that they are different across species, among species, only because these species have different size. I mean, not only. They are different because species have different size. The trabecular structure of an elephant is different from the trabecular structure of a mouse, not only because they may have different locomotor behaviors, but also because they are drastically different in size. So we should somehow be able to purify, let's say, to clean the ecological functional signal from the allometric one. And similarly, as I already said before, we should always remember that species have differences or similarities only because they have shared, to different extents, shared evolutionary history. So it is another signal that we should try somehow to remove or to clean or to understand, to quantify, and to separate from the ecological, so from the purely ecological one. And it is not easy, of course. So all of this has been used in my PhD, which was undertaken at the Humboldt University in Berlin, in Germany, and in 2022. So basically, as a case study, I addressed a lifestyle, so an ecological adaptation evolved several times in mammals, and the evolutionary process on which I focused is the process of convergent evolution or convergence, which in brief can be summarized as the independent evolution of the same phenotype. So not necessarily morphology. Think about ecological adaptations, for instance. So that the textbook example of convergent evolution is the independent evolution of flight occurring in bats and in birds, for instance. We know that bats are mammals, birds are not mammals, but they evolve the same ecological adaptation. It occurred independently, so with 2 independent events in the evolutionary history, and this process is called convergent evolution. The thing is, I identified these 7 events of independent evolution of the same lifestyle, which is the lifestyle called slow. So the lifestyle that we can call slow arboreality. So basically, I focused on slow arboreal mammals because I studied mammals, and we can define this adaptation as the adaptation as adaptation in which we have species with a particularly cautious arboreal climbing behavior, a life balance dominated by rest and patience, and with an extremely low metabolic rate. So it occurred at least 7 times in mammals in the 2 lineages of tree sloths that, as you may have heard, although they are extraordinarily similar, it is now well established that they are the result of 2 independent events of evolution. So basically 2 times independently in the group where they belong, called Xenarthrans, together with anteaters and armadillos. They evolved 2 times independently with the same peculiar lifestyle, but it occurred another time probably in Xenarthrans with the silk anteater, this peculiar small anteater. We have, of course, the famous koala. And in primates, we have lorisids, which are known for their stealthy, slow, cautious lifestyle. And it occurred probably 2 other times in 2 extinct taxa, primate taxa, that are the so-called sloth lemurs and koala lemurs. That's the name that say. You can understand that these species, they are strepsirrhines, so they are close to lemurs. They were large, particularly large, but we know today that probably they have a very slow arboreal lifestyle. So the thing was trying to understand if 2 limb bones, I basically focused on the humerus and the femur, can show morphological similarities mirroring the ecological similarities. So basically I focused on ecomorphological convergence. We know that there is this convergence of ecological traits and characteristics. I wanted to see if the morphology of postcranial long limb elements, those that I mentioned before, responded to these independent events of convergence of ecological aspects. So the first trait I focused on is this trait. This is the microstructural trait I mentioned before that can be appreciated with very high resolution micro-CT scans. It's called cortical compactness, as the name clearly says. It basically informs on how much the cortex, so the external compact layer along the central shaft that I mentioned before, how much is compact. And of course, the more the cortex is filled with these vacuities, the less it is compact. So we knew before starting the work that previously it had been shown that tree sloths, so just for you to remember, the 2 independent lineages of tree sloths, so the 2 convergently slow arboreal lineages of tree sloths, were shown to have a peculiarly low, particularly low cortical compactness. So for some reason, in tree sloths cortex, we have like this widespread distribution of vacuities, and it is different from all the other mammals. So it was like a peculiar condition which increase the curiosity on this taxa. And the hypothesis was that since they are, again, convergently evolving their lifestyle, their slow arboreal lifestyle, it was hypothesized that this trait, this morphological trait, may be a functional trait. So it may be something to do, it may relate somehow to their ecological adaptation. So the question was, starting from this previous evidence, the question was, if it is true, it is mirrored by other slow arboreal mammals too, because if it is true for tree sloths, I can expect to have the same condition in other slow arboreal mammals. So there is a significant difference in cortical compactness between slow arboreal mammals and all the other taxa. So we can, and it is for a low cortical compactness as we were expecting. The thing is that we didn't find this condition in all the 7 lineages that I mentioned before, but only in some of them. So it was confirmed in tree sloths, as we were expecting. It was found in the silk anteater, so the small anteater that, as I told you, evolved the same lifestyle. And it was found also in the 2 lineages of extinct primates, so the sloth lemurs and the koala lemurs, but it wasn't found, for instance, in the koala and in lorisids. So basically highlighting the complexity of the phenomenon to try to understand which is the functional explanation for this trait. We basically posited 2 potential explanations. The first one is that if you compare the CT scans thin sections with thin sections, the CT scan 2D sections with thin sections, so histological sections, you can somehow identify these vacuities as resorption cavities that are related to the continuously acting process of bone remodeling, which acts during life, basically till death. So since this process is a systemic process happening in all the body, we hypothesize that it may be something to do with the low metabolism of this species. Just for you to know, the tree sloth, one of the two tree sloths, the three-toed tree sloth, is the mammal with the slowest metabolism. Most are very, very hypometabolic. Another potential explanation is strictly biomechanical, because we know that these taxa, because of their suspensory behavior, they tend to have their bones in tension. And when a bone is under tension, it's called that the diaphysis has an increased compliance. And this property basically involves increased microcracks. So we can have also a biomechanical explanation to justify these peculiar characteristics in the tree sloth cortex. So for the second and the third papers of my works of my PhD, I focused on several levels altogether. So again, I focused on the diaphysis, but not at so high resolution, more with a cross-sectional property approach, the one I mentioned before, showing you the beam. I also focused on the extremities, on the epiphysis, sorry, and also I focused on external shape, so the general morphology that we more commonly study through, for instance, 3D genetic morphometrics, because I wanted to have an integrative approach. Again, the question was trying to understand if the morphology of these low arboreal mammals follows what happened to their ecology. So if they evolve independently, morphological similarities following ecological similarities. The first work was only focusing on a subgroup, which is the one I already mentioned before, the one called Xanarthrans, because the thing is that Xanarthrans offer an opportunity, because we have the two lineages of tree sloths, as I told you, they are extraordinarily similar. They evolve this similar lifestyle independently. But the silk anthider, which is another slow arboreal mammal, is more similar to them than to the other anthiders, but not to the same extent. You can see the thickness of the arrows to try to convey this message. So basically, we have two lineages that are extraordinarily similar and convergent, and another lineage that converge to them somehow, but less intensively. Okay, so these different nuances of convergence were called complete convergence in the case of extraordinarily similar traits, as in this case, the ecological one, and it is called incomplete convergence in cases in which they have some similarities, but still they retain some differences. So you can think about different degrees, different intensity of convergence. Here I will not go into detail, but the thing is that you can identify these different nuances studying the angles of the trajectories, bringing them to invade some spaces of some areas of the morphospace. So I need to very briefly define what is a morphospace. Probably someone of you already knows what is it. Basically, a morphospace is a space where a space or a scatter plot, a biped, a B-plot, where you have some points, and the position of each point on the plot defines the morphology of a single taxa. So the closer two taxa are on the morphospace, the more they are similar. Basically, this is the only information you should keep in mind. And if you map the phylogenetic tree, so the phylogeny on the morphospace, you can also include phylogenetic information. So you can see, for instance, if two taxa tend to depart and invade the same region of the morphospace. So basically, if they tend to evolve the same morphology, a similar morphology or not, because in this case, for instance, they can go in different directions. Specifically for this work, I wanted to see if the taxa are convergent. So translating convergence into morphospace patterns, it means that two taxa independently start from different points and go to invade the same areas. That means becoming more similar, since the morphospace informs on this. I was basically able to see that tree sloths are more similar, so they are more overlapping, they are closer on the morphospace differently from the silk anthider, so basically confirming the pattern I hypothesized before of complete and incomplete convergence. And I was also able to quantify these patterns. Here again, I will not go in details, but basically there are some indices that you can use, you can quantify with an R package. These indices are very straightforward and easy to be interpreted, because they tells you how much taxa tend to be closer on morphospaces. So the higher is the index, the closer they are on the morphospace, and vice versa, of course. And quantifying this index, again, we were able to see that tree slots tend to be more similar in morphology, mirroring what it happens to their ecology differently from what each of the two tree slots does with the silken cedar. I hope I was clear, but I'm not sure. Okay, so the thing was, the important thing of this work was also to see that especially the internal bone traits contribute to this pattern. So again, we come back to bone inner structure, especially bone inner traits are responsible for this pattern. And so it leads to the last question of my PhD, which was trying to understand if inner bone converge more strongly than external shape. Okay, I know this question is weird, so I will try to explain it better using a theoretical framework. So we can think about this taxa, as I told you, that independently evolve the same ecology. Okay, and every time it happens, following the more convergent evolution expectations, if morphology follows ecology, we can think about morphological similar traits, no? So similar morphological traits, sorry. So my point was, since this pattern of complete and incomplete convergence is mainly mainly explained by inner traits, is it possible that when two lineages independently invade the same ecological niche, the inner traits respond more compared to external shapes? So in other words, is it possible that inner traits changes more quickly and more intensively compared to what it happens to external shape? So here, without going too much into details, we know that every time the phenotype evolves and changes along evolutionary history, it is due both to intrinsic factors, which can be genetics, phylogeny, ontogeny, and everything related to the internal characteristics of the species, or extrinsic factors, which are basically how the species interact with the environment. So we can say ecology in a word. So it is known that the intrinsic factors, okay, so if we only think about phylogenetic history, genetic characteristics, ontogeny, so everything inside the species, okay, inside the species characteristics, leads to an emerging property called evolvability. When we talk about evolvability, without going too much into detail, we should think about this definition. That is the propensity to evolve in response to opportunities. What does it mean? If you think about a morphospace, so the same plot I showed you before, every time a species evolve an ecological adaptation, so baits an ecological niche, it may change morphologically accordingly. The more it changes, the more the morphology is evolvable, basically. So the larger is the area of the morphospace that is invaded by the species, the higher is the evolvability of a specific trait. So we can think about that a trait is more evolvable if, starting from the common ancestor, it goes and invades more space. So you can see here, for instance, that the red and the orange clades are more evolvable compared to the blue, no? Because you see they depart more from the common ancestor. This is the only important concept at this stage. So if we think about a process in which the extrinsic factors are kept constant, okay, so we think about similar ecological conditions, basically, okay, we can say that everything, all the difference that we appreciate are due to intrinsic factors. It is like a treatment control experiment, no? You basically put two observations, in our case species, in the same condition, in the same ecological condition. All the difference that I appreciate between these two species should be due necessarily to intrinsic factors. Of course, it is not possible to really control, because we are talking about evolution, which is not repeatable, but it is what can be called a common garden experiment. This experimental setting is widely used in botany, for instance. You can take several plant species and put them at same environmental conditions, and any difference that I appreciate between the plant species should be explained in terms of intrinsic characteristics of the plants. Of course, again, it is different. This is purely theoretical in our case because we are talking about evolution. But if you think about convergence, it's basically allows us to follow this theoretical framework, because every time a species enter every time species evolve independently, the same ecological adaptations, they are like in the same ecological environmental conditions, and so they are like, we are kind of controlling the extrinsic factors. So any difference that we appreciate between these lineages should be due to intrinsic factors, and as I told you before, intrinsic factors mean evolvability. So all of this is to say that it allowed me and allowed us, me and my collaborators, to see how internal structure, so trabecular bone, diaphyseal structure, blah, 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 evolve compared to external shape using the indices of convergence strength that I mentioned before. So basically, we can measure how strongly internal structure converge and how strongly external shape converge across these taxa, and we can compare the convergence strength. I'm not sure, again, it was clear. But the thing is that if you measure this convergence strength in two different anatomical levels, internal structure versus external shape, using these indices, it is pretty clear that the internal structure converges way strongly, more strongly. So basically confirming our expectation, which was based on these, expectations that are basically based on anatomical constraints that I can tell you more later if you want in the questions. Question and answers. And the phylomorphospace, so the morphospace, this plot I mentioned before, again, show, as you can see, again, the convergent taxa, as I told you before, they independently tend to invade the same morphospace area, no? So basically allowing let's back, okay? Allowing us to have a theoretical confirmation, practical confirmation of the theoretical definition of convergent evolution. And the traits mainly responsible for this pattern are internal structural traits, so confirming what I was expecting. Oh, thank you. Okay, finally, I will briefly tell you something on my current project on birds. So basically, all these concepts and the principles I mentioned so far are used in my current project, my postdoc project, which focuses on birds. So basically, I study only the wing bones, so the humerus and the ulna, at the levels I mentioned before, so the epiphyses and diaphyses, only the internal structure, because I would like to see which traits are mainly affected by the factors I mentioned before, so the ecology, allometry, or phylogeny. So when I find, as I told you before, a relationship between morphological traits and ecology, I can try to infer the paleobiology on some fossil birds. And also another axis of the project would like to see if ecological driven traits are already present at birth. So when a bird is after the bird's birth, basically, so post-hatchling, and which ones instead need to be developed along ontogeny. I think I took so long, so I will not be so detailed in this part. But basically, okay, I can say that in general, I decided to study birds because they are particularly biodiverse taxon, so they can be considered like a didactical case study to understand how these factors affect the morphology. And of course, I studied the wing bones because the flight is crucial, as you can easily understand in the evolution of the entire clade. And so far, these bones were mainly studied in outer morphology, so in the external shape. But similar analysis of the internal structure are rare. And as I told you at the beginning of the talk, this level actually allows us to understand how different factors as ecology, phylogeny, and ontogeny affect the evolution. So I'm studying around 200 species of birds, ideally covering the entire diversity of the group. Of course, there are 10K species, so it is possible to study all of them, but the coverage should be more or less uniform. And I'm studying the humerus and the ulna of all these species through micro-CT scans. Many of them are already available on Morphosource in the Bird Tempo project, and additional ones were collected in the Museum of Zoology in Cambridge and the Natural History Museum in London. So basically, after you rend the bone in anatomical standard condition, directions, you extract I told you before, both the epiphyseal trabecular bone with this machine learning approach, allowing to study the entire epiphysis, and the diaphysis, the central shaft, allowing to have around 40 structural traits per bone. To quantify phylogenetic effects, of course, you should use a phylogeny and then using this one. To study allometric effects, you should have data on body size, and I will take them from a database. And to study the ecological effects, you should try to characterize the ecology. And basically, I'm taking data on habitat, primary lifestyle, and flight style. So to take into account different aspects of their ecology. And doing this through a cluster analysis, it is possible to identify different clusters, ecological clusters, different ecological clusters that allow to appreciate ecological diversity. So in short, in brief, basically what I found so far is still a preliminary analysis, so I only collected data on the 25% of the species, is that if you see the distribution of the species on the morphospace, they tend to cluster all together. So it seems to be not a so strong ecological effect, except for some extreme adaptations as penguins and guillemots or hummingbirds that seems to have a particularly divergent morphology. I will not add anything else here. If you want to know more, I can tell you later. But basically, in short, I can tell you that it mirrors what was previously found studying the outer shape. So there seems to be a correspondence between what is present in the external morphology of the wing bones and what is present in the internal morphology. But again, it is only a preliminary result, so we'll try to find more in the complete analysis. So I'm sorry for being so long, but if you want to know more, you can ask me later. Thank you for your attention.